Excellent. Good afternoon. My name is Tim Morris. I'm the Chief Financial and Chief Operating Officer for Humanigen, here to touch a little bit on the development we have with our primary molecule, which is linzilimab. It's a monoclonal antibody that is an anti-GM-CSF molecule. It's been progressed more recently into the COVID-19 space, so let me touch on that a little bit. As we heard from the last speaker, we think COVID is here to stay. It's very similar to influenza. We do think it will be seasonal. Hospitalizations will continue to occur both in the U.S. and in the European Union. Deaths are high, despite nice numbers of vaccination rates and improvements to standard of care. We think the opportunity here is going to be large from a commercial standpoint. Just by way of reference, remdesivir sold about $5 billion last year. Actemra sold about $2 billion last year. So there's clearly plenty of money to be made in the COVID space. We do reference the influenza market here because we think this is a good example of what can happen when some of these viruses will mutate despite having vaccines that could work. Those efficacies from the vaccines will wane, and escape variants will emerge from this. So we're currently planning to have a decent amount of hospitalizations in the U.S. and in the UK and in the EU as the primary markets that we're going to focus on. We think that there is a need for additional COVID-19 treatments. Our product is hospitalized for patients who are hypoxic. What we've seen here, remdesivir is an antiviral. We think the efficacy is probably questionable at best. Steroids have probably minimal impact on low flow and room air patients. Toci, again, is another one that's been involved in very large studies, but I think the data is somewhat questionable. While patients can prevent going to the hospital through vaccines or through oral antivirals, we think some of those will progress. And what we're trying to do for patients is we're trying to prevent them from either dying or getting on mechanical ventilation. And so that's really the positioning that we think linzilimab will follow. A little bit of science here. What is GM-CSF? It kind of sits on the top of the cytokine cascade. It causes, if left unchecked, eventually lung damage and tissue damage here. The body's overreaction to the virus is what's going to cause death here. It's not the virus itself. So, and the literature of the last couple of years about the value of GM-CSF as a target, I think is relatively strong. We've also spent some time and effort looking at CRP as a measure of inflammation, and we think we've kind of narrowed down a patient population that should respond a lot better to linzilimab than just the general population. So linzilimab itself, as I mentioned, is going to be targeting GM-CSF. This is going to be different than the drugs that target IL-1, IL-6, and we think that it's something that has a high affinity here, high binding. The criteria for our study is that patients will come in if your oxygen saturation level is below 94. We think you're a good candidate. We do treat patients who are in room air, low flow, and high flow. Again, it's a single-day treatment. It's given 3 times. It's 1,800 milligrams here, given IV. We've studied this on top of standard of care, which is going to be steroids, which is going to be remdesivir, and a variety of other treatments. And more importantly, we are variant agnostic. And so again, it's as not an antiviral, it's an immunomodulator. Whatever variants come, we should have some efficacy against that. From a positioning standpoint, there was a workshop held in the UK from NICE, and so where we've kind of positioned linzilimab is right along with their positioning. Obviously, you can get oral antivirals to prevent infection. You can clearly get vaccines, and at the back end of the spectrum, you are able to get baricitinib or tocilizumab for folks that are ventilated here. But where we think linzilimab should be positioned is folks who come into the hospital, they have a oxygen saturation below 94, and probably more importantly have a CRP level, which is a measure of inflammation, below 150. Average for just walking around is about 5. And so but we had patients as high as 300 in the CRP level. So this is where we think the product should be used. The results from the first phase 3 study that we ran were published in Lancet Respiratory Medicine. I'll show you those data in a second, but this was a phase 3 study that was run by ourselves. The endpoint here was looking for survival without ventilation as compared to standard of care. The trial design itself was looking, again, measuring patients' assessments over day 28 with a 60-day follow-up. We did measure a number of secondary endpoints here that helped. The study really wasn't powered. It only had 500 patients, so you don't get, while you get trends towards mortality and reductions in time to recovery, it really wasn't powered for that. So we did hit the primary endpoint here, which was ventilator-free survival. Oops. It doesn't like that one. It wants to jump. Sorry. So you can see here the linzelumab group, 84% of these patients did survive without a ventilator as compared to the standard of care here, which was about 77%. And while the absolute numbers aren't that large, you look at it as a hazard ratio, so 1.54. That means you have a 54% relative risk, better chance of survival without ventilation with linzelumab than without it. Now, this was kind of an all-comer study. We didn't do any work ahead of time to try to segregate patients, but we've done work on a post-hoc basis to look at what patients it really works best in. And what we found is that patients that have CRP levels less than 150. And so why that's important here, a couple of things. One, you can see the p-value goes from 0.04 to out 4 digits. Hazard ratio goes from 1.54 to 3.04. And you can see that the improvement in the absolute and the relative risk basis gets much better. Why this is important is because we are running a second study sponsored by the NIH. And when we saw this data that came from LiveAir, we went to the NIH, and so they've modified the protocol to look at exclusion criteria for CRP level patients above 150. And probably more importantly, the statistical analysis plan is now going to look at this as the primary patient population. So we believe this trial is designed for success. This identifies the set of patients that it works best in. And while this is on an ad hoc basis, you can see that the data is very strong. We were named as one of the top 10 drugs in development in the European Union here. We think that that's very helpful. Unfortunately, it hasn't helped with the regulators just yet. We do continue to work with the EMA and we'll file for approval most likely after the ACTIV-5 study reads out. This is the ACTIV-5 trial right now. It's called ACTIV-5 Bet-B. It's a study run by the NIH, and we were selected to be the only GM-CSF inhibitor to run in one of these studies. They're looking at everything against a background of remdesivir, which is fine. We seem to have some synergistic efficacy here. The primary endpoint, again, is aligned with the LiveAir. You're going to look for patients that are alive and not on a ventilator through day 29. And again, looking at the CRP patient population that are below 150. So we also think that the NIH support is going to be helpful in dealing with the agency and working with them to try to get the product approved under emergency use. Oops. This is a lot of data on this slide, but historically there are a couple of immunomodulators that have been approved. I mean, dexamethasone is a steroid, and toci, as I mentioned. When you look at the recovery study, which was probably the largest study that was done with these patients and thousands of patients, the relative risk here is relatively small. And so for Dex, it's about 8%. For toci, it's about 15%. Now, clearly not a head-to-head comparison here, but the goal for ACTIV-5 is to see if we could take and probably beat toci by 2x. And so looking at, on a retrospective basis, where you can see linzelumab was able to have a relative risk reduction of about 33%, we think that that's a win. So if we can come in on top of steroids and try to beat toci, we think that that's what our expectations are. So our public guidance is that we will have results from this study by the end of this quarter. We've also done some pharmacoeconomic work here. I mean, as a monoclonal, it's not cheap to make and it won't be cheap to buy. But when you take a look at some of the pharmacoeconomic outcomes that come, particularly from this set of patients, whether it's reduction in ventilators, days in the hospital, ICU days, or numbers needed to treat, that it adds up to about 8,000 pounds of savings. At least these are numbers based on the UK system. We've done the equivalent in the US, and it again equates to about 13,000. So we think that the pharmacoeconomic work will be helpful, and as we go ahead and seek reimbursement for this, we should be able to get reimbursed at a fairly decent level. These are just some of the data that we've used here. You can see nice reduction in whether it's ICU days, total bed days in the hospital, or mechanical ventilation days. So all very nice reduction of costs and savings, not to mention saving lives. Where we are from a regulatory standpoint, as I mentioned, this will cascade nicely with the readout of the Active-5 data. We have had a pre-EUA meeting with the agency. They've agreed to the statistical analysis plan and the data that we'll include. We should be in a position to file the EUA shortly after our top-line data. In the case of MHRA, we are on a rolling review. We've had multiple meetings with a variety of different groups there. They were able to give us some feedback in the fall that we've been working on. Those will be ready to submit alongside the data that we get from Active-5. And then in terms of the European Union, we have had rapporteur and co-rapporteurs appointed, and we've been given a time to submit sometime in September. So I think it actually will fall in nicely. We do have the product available through a named patient basis in certain countries in Europe as well, and looking at trying to do maybe some local filings for other countries in Europe. This is the last slide here. We do have a number of other products, other indications that we're working on that are not COVID related but have to do with cytokine storm. The first one is the SHIELD study, which we're going to be doing alongside of CAR-T therapy, essentially Yescarta. This was shown in a very small study to be very effective in lowering cytokine storm and neurotoxicity. So we're looking to dose our first patients here, by the mid-year. The second study is going to be a study called the rating study, which is going to be run by a group in the UK called the Impact Partnership. This is going to be in graft versus host disease. And again, that study should enroll the first patients here by mid-year. And then the last study, it's a little bit of an outlier. It's pre-CHIM. It's chronic myelomonocytic leukemia. This actually what happens when you have too much production of myeloid cells. So it's a very rare blood disorder. Here it's being run down in Australia at a couple of centers there. The compound was actually invented and discovered there. And so early data looks really nice in this set of patients, and we want to continue to expand this and probably expand the study. So I think with that, I will end the presentation. I thank you for your time and attention, and stay tuned for some data readouts, hopefully later this quarter.
Thanks very much, Tim. Any questions?
Excellent.
Everything perfect.
All good. Thank you, guys.
Thanks very much.